weights down, beg forgiveness, crash the known way. Father, he enjoyed courageous, others walked away. A snowflake falls in rain. Any doors are open now, as the bells are ringing now, 'cause the man of the hour is taken his final bow back for. Nature has its own religion, gospel from my long division, young man may pretend, old man comprehend. And the sky breaks at dawn, spreading light upon this town. We all come back, 'cause the man of the hour is final bow back for. The old man broken and free, long resting. It's final bow as the curtain comes down. I feel that this is just life.
Wanking. Not balls. My favourite piece of the week. What do we call this, Bev?
This is edible egg share. Just 40 payments of $19.99.
Gorgeous.
It's just so lovely. I want to buy one for myself.
You can't afford him.
Ricky can.
He's not for sale, Bev.
Can I be last right? What?
Eat the damn fry, Ed. You can have all the fries.
Come on. Oh.
Welcome to Metabolic Physiology in Isolation, where today we're going to hear from Isaac Harris. Yep, we're starting in 5, 4, 3, 2, 1. Welcome, welcome, welcome. Glad to have you all back. We talked about this yesterday, so I'm not going to go through it deeply again today, but I do really think that we're launching what can be the new model for global inclusive science. It's cost effective. You all can listen to this anytime you want, any way you like, for free. White McGarrett Lab are very happy to take on the small cost it takes to launch this. I'm really enjoying the Twitter space and all the things that are being shown. I know everyone is still super impressed by Cassidy. Right now it's your turn. We really liked the princess hat yesterday. I think she's got the MetPhys 2020 prize for the best princess hat. I do like the blue and pink hair. I wish I had something like that for myself. I've come back to a post from Gustavo Sean. I really do think this is the most poetic summary of somebody's talk. Are you lost, she asked, to what he replies, no, of course not. Well, he takes another reluctant look at the map. I think some of us sometimes feel like that sitting through talks, but I hope we all got a lot out of this. Michael Nugent, enjoyed the soundtrack yesterday. Thank you so much for the feedback. I spend about a minute a day thinking about what we can put into these, and I've decided based on that one positive response, when I figure this out, we're going to have a Spotify playlist for Metabolic Physiology in Isolation. All the tunes we've played beforehand are going to be in that list, so you can enjoy them while you are isolated. I've said this every day. I'm going to reiterate it again today. Please take note of the speakers. These are the wonderful people who have agreed to come on, give their time, give a talk, share their unpublished data, their published data in a personal way, so that you all can enjoy the cutting edge metabolism science from your home. I know you're all enjoying it. I see a lot of questions online. Keep it up. That's really means a lot to the speakers. Those Facebook and YouTube icons show where the recordings are available. If you go to the White McGarrett Lab Facebook page, you'll be able to see the recordings from the live stream. If you go to YouTube and type hashtag MetPhys2020, you'll see all the talks coming up. By this afternoon, we'll have Isaac's up. If you have friends that are interested in learning this, if you want to share with students or colleagues, feel free, let them know. This is going to be there for everyone to enjoy, and I think all the speakers are happy if you share. So please share broadly. Like I mentioned yesterday, there's a new list of speakers that get us out to April 24th. That is where MetPhys 2020 will end. The good news is we'll be back for MetPhys 2021, and we're already developing a really great speaker lineup with the same model. I think sharing cutting edge science at a global scale where everyone can listen to it and see it without requiring any cost is really the way to go for the future, and we're happy to keep extending that. If you do see the speakers in public, let them know how much you appreciate their talk and their willingness to share their data. That's about all I need to say about that. If you want to thank me, the way that would really honor me is if you made a small donation to the Pathway to Stop Diabetes program. Myself and also Salim Riera have been supported by this program. And if you look on that website, diabetes.org/pathway, you'll see all the fantastic science that program is supporting in particular. So that's how you can thank me, but you don't need to, just enjoy the talks. And tomorrow, please join us again. Tomorrow's going to be a fantastic talk from one of my favorite scientists. She's a very funny individual, and I really enjoy listening to her talk, and I'm sure you will tomorrow. She's going to present, Carolyn Cummings is going to present LXR-beta's role in glucocorticoid-induced diabetes. So please join us tomorrow, last talk of the week, and it's going to be a goodie. All right, so I'm going to hand the controls over to Isaac now. Enjoy, everybody. Making you the host, Isaac.
Can everyone hear me? Actually, maybe I won't be able to get a response from that. Let me share my screen. Oh, I have confirmation that I can be heard and seen. So first, I'd just like to thank Philip and for organizing this, and everyone for taking the time out of their day to tune in. I think this is a really great opportunity. I also like to give really a shout out to all the women and men across the world, you know, obviously the doctors, nurses, and everyone involved in trying to help all the patients with regards to COVID-19, you know, yeah, you know, this definitely is a different scenario, but I'm excited that we can still participate in science going forward. So as Philip mentioned, my name's Isaac Harris. The title of my talk is Understanding the Roles of Antioxidants in Cancer. Now, antioxidants, this is a term that pretty much everyone and even their family members probably outside of science have heard this term before. It's in the media, everyone talks about it. You know, antioxidants, they're really good for you. They're this kind of cure-all for almost everything, and even cancer. You know, it's been long postulated that antioxidants can have a preventative effect towards cancer. But what I've found over the course of my research career is that you know there's really a lot of questions that remain to be solved with regards to what exactly these antioxidants are doing and what their roles are in normal physiology and in disease physiology such as cancer. So this is really the crux of my talk, is understanding these roles and trying to take this further towards therapeutics. So if we get going, the first thing we have to look at is, what do these antioxidants do? And so what antioxidant, you know, it's in the word, it's an antioxidant. It's something that quenches or buffers reactive oxygen species. And I think one of, you know, the confounding factors or difficulties in fully interpreting all the effects of antioxidants is that the level of reactive oxygen species that is present in the cell greatly determines its overall effect on the cell. I don't know if you can see my pointer here, but at low levels, you can get really reactive oxygen species ROS acting as a secondary messenger towards just cell proliferation, cell signaling. At higher levels, you can get damage to DNA, mutagenesis, and potentially transforming events. But if this ROS is allowed to build up to such a high level, this really becomes a stress. It becomes an oxidative stress. And this is ultimately very detrimental to the cell, and it can lead to cellular senescence or ultimately cell death. Now, I believe a large proportion of scientific literature over the years, and especially in the public domain, has focused on this intermediate role, where if ROS is able to build up to the point that it's inducing mutagenesis, you know, it would logically make sense that if you had a normal cell and you were able to increase antioxidants, you could push it out of this mutagenic range, potentially preventing transforming events and preventing cancer. This situation, though, changes dramatically once a normal cell is a neoplastic cell or even a pre-neoplastic cell. And this is really ingrained in, well, where are the source of ROS in the cell? You know, where is this reactive oxygen species coming from? And we know in our literature that it's really a byproduct of normal cellular processes. This includes mitochondrial metabolism, also protein synthesis and translation, and there's probably a lot of other cellular pathways that contribute to increased reactive oxygen species in the cell that we don't fully appreciate yet. But what we do know is that, like several studies have shown over and over again that normal cellular processes are upregulated to overcome cell death pathways and ultimately to keep growing. And so a cancer cell is actually upregulating its metabolism, upregulating its protein synthesis, and therefore, just based on a pure numbers, it's hypothesized that it's producing more of this reactive oxygen species as a byproduct. And so now this reactive oxygen species becomes a stress, and it's very much a limiting stress where the cancer cell, again, hypothetically would be dependent on increasing these antioxidants to push it out of this detrimental range. And now it becomes that the cancer cell itself is relying on these antioxidants to survive. So, I've mentioned there's obviously a lot of talk about antioxidants in the public domain as a supplement, but what sort of actual scientific data do we have behind this? And really, there is quite a bit, but one of the most largest study, really, and one of the most impactful was a study that came out quite a few years ago now, and it was started even longer before, and a bunch of clinicians and doctors and scientists said, okay, let's actually put some science behind this theory that antioxidants can prevent cancer. Supplement industry is a completely unregulated industry. It doesn't need FDA approval, or sorry, it doesn't need FDA approval. You're selling these supplements, but what are the actual effects on this? And so this was at the time the largest cancer prevention trial ever undertaken, more than 400 clinical cancer centers really across the world. And they were looking at if they supplemented with different antioxidants and measured probability of tumor incidence, what would happen. And unfortunately, the worst that could happen did in fact happen. And instead of actually preventing cancer, they were finding a statistically significant increase in tumor incidence in the patients actually receiving the antioxidant supplement. And so this is this vitamin E. And again, what is, was pointed out to me by quite a few people that what's actually pretty scary is, this wasn't, they didn't just pick some absurdly high dose. They actually used doses that is used by the public, and I think it's even reported up to 10% of them, Americans take these sort of supplements. And so, this was quite concerning. And so, what our lab really focuses on is the opposite, is trying to understand, outside of supplements, how does the cell make its own supplements, make its own antioxidants? And if we were to block these processes, would we be able to have a therapeutic effect on diseases such as cancer? So to do this, and really what ties into the metabolism aspect of this seminar series, is that we have focused our research on glutathione. So this is the most abundant antioxidant cofactor in the cell. It's one of the most abundant metabolites in the cell. Excuse me. And what's really interesting is that it's only made of 3 amino acids: glutamate, cysteine, and glycine. And unlike numerous other metabolic pathways, lots of us have seen these huge roadmaps with, looking like some sort of traffic jam where there's multiple feedbacks and redundancies and multiple different ways to get metabolites and metabolic precursors and stuff like ATP and nucleotides and stuff like this. Glutathione doesn't have that. There's only, to what at least has been elucidated to date, a singular enzyme that drives the de novo synthesis of glutathione. And that's this glutamate cysteine ligase, and this is the rate limiting step of GCLC. Now there is other enzymes that contribute to these processes, one being GCLM. It has no catalytic activity, but it greatly increases the rate of reaction. It also allows for a larger buildup of glutathione, preventing its negative inhibition. And so really these two enzymes and using other chemicals, we've tried to focus on asking very, very naive basic questions of, you know, what are the roles of this antioxidant glutathione in other, you know, in physiology and disease. And so to interrogate this, and what I'll be presenting on today is, you know, you can delete this modifying unit. And so here you still have the catalytic activity, but it's just happening at a much lower rate. You can use a well-established chemical compound, butyl sulfoxamine, or BSO. This binds to the catalytic pocket and basically poisons the reaction to stop GCLC from carrying out its glutathione synthesis. And ultimately, you can delete the catalytic unit. And again, to date, there has been, at least in mammalian cells, no redundancies that have been found to overcome loss of GCLC and glutathione synthesis. And so this process has been the focus of, to a large part, my research career and is the focus of our lab going forward. We've really tried to hone in on this to ask numerous questions of the roles of antioxidants in cancer. We've asked this with regards to initiation, progression, also drug sensitivities, and I'll touch on that a bit later. And ultimately, what is happening for, you know, what are the roles of glutathione in normal cells? Can we tease this apart? How much is correlation versus causation? And can we use different tool systems to better understand this? So just to get going, so all this work started during my graduate studies. I was in the lab with Dr. Tak Mak at the University of Toronto. There was, I want to say in the early 2000s, this boom in cancer metabolism, really seminal studies from Matt Vanderheiden, Luke Cantley, Craig Thompson, and numerous others, really focusing on metabolism and energy requirements for cancer cells. And in our lab, we started to, in Tak's lab, he was starting to think of other necessities, and one being, okay, what about dealing with the oxidative stress that comes with energy generation? And along this time, there was also really, really great papers from Joan Brugge's group, Zach Schaefer that spoke earlier in the seminar series, Gina De Nicola and Dave Tuvesen's work, Michael Clark, all showing very important requirements of antioxidants for tumorigenesis and cancer cell survival. And so all this kind of came together, and you know, when I was grad school, I was like, okay, how can we interrogate this? How can we do really even the opposite of this clinical trial? How can we, instead of supplementing with antioxidants, have a system where there's less antioxidant production? And so we turned to this mouse model for the knockout of the modifying unit, GCLM. So again, as I mentioned, GCLM has no catalytic activity, but it aids in the process. And so when you lose it, these mice have a reduced production of glutathione, but they're actually completely embryonically viable. There is certain phenotypes with them. They have fertility issues. They're sensitive to drug-induced liver toxicity, but they have a normal lifespan to what has been reported. And we really thought this was a great model to interrogate, to ask, again, a very naive question. Can we, you know, if you have the opposite of supplementation, if you have a systemic reduction in glutathione synthesis, what would happen in cancer? The cancer model we turned to was breast cancer tumor model. This is the MMTV mouse mammary tumor virus driven PYMT breast cancer model. And so these mice get quite aggressive, rapid, multifocal breast tumors. And again, we just took a very naive approach in saying, if we take a very aggressive cancer in vivo spontaneous tumor model, what would happen? And very much in line with the clinical trial, here we were finding that when there is actually a reduction in glutathione synthesis and antioxidant production, you're actually getting a delay in tumor onset. And so here measuring tumor-free survival over time, and all this work is published quite a few years ago now. In our knockout mice, we get a very significant delay on tumor onset. If we take this further and actually look, is there a difference in final tumor burden? There is. These tumors are much smaller. And what was quite interesting was, what they looked like under the microscope, and looking at the histology and showing this to pathologists, it was told to me that these were largely benign tumors that was left at the very end. And so you can see a very typical PYMT breast tumor, invasive ductal carcinoma, whereas the knockout, there's very few cells, there's even normal architecture of the mammary gland, and this is just with an H&E stain. If we measure proliferation with a Ki-67 stain, you can see these are largely non-proliferative cells. So this was, really exciting. We got really lucky and with, this experiment, and we were able to see this effect. But we were also left with the question being, how could this work as an intervention? Stopping breast cancer from arising in a mouse model with a deletion of GCLM, that's not quite as physiological as we wanted. So, there was definitely some questions with regards to, okay, what if we block glutathione synthesis at different stages during the course of tumor development? And so to do this, we turned to a chemical approach, a chemical inhibition approach, and we use this chemical compound BSO, or butyl sulfoxamine, as I mentioned. And so using this chemical compound, we dosed the mice at two different stages. We dosed them prior to tumor onset, so 4 weeks after birth, just after weaning, we started them on this treatment, similar to the GCL knockout. BSO treatment reduces glutathione synthesis. It doesn't fully ablate it, and I'll touch on that a bit later. But these mice are totally fine. And it was really encouraging to see now, if we measure tumor mass using BSO at this early time point, there was a very significant reduction in tumor mass and a very large difference in histology. But if we waited until the tumors had already developed and were really going, we saw really little or no effect. And so here, when measuring the tumor mass, we're getting a non-significant difference, and under histology, again, they look very similar. So this left us kind of scratching our heads with regards to what next, what are the next steps. We had seen that it appeared that glutathione, systemic glutathione synthesis at its highest level was required for cancer initiation, or the early stages of cancer initiation. But at later stages during cancer progression, it was potentially not required. And so this made us go back to the drawing board and say, okay, what else is there that we could be missing? And obviously people that research antioxidants, they would say, there's a whole lot missing. And so we started to say, okay, maybe we're looking at this too myopically of just looking at glutathione. Is there other antioxidants involved? And so we and others had actually seen that this protein thyrodoxin, which again is protein synthesis limited, and so it's a protein that is very efficient at detoxifying reactive oxygen species, and it's governed by its regeneration by NADPH. But here, if you actually blocked both glutathione synthesis and this thyrodoxine pathway, well, you would have an overwhelming amount of reactive oxygen species and ultimately cancer cell death. Now, just to give you some time frame, this was at, you know, I had finished my PhD. I'd moved to Boston to work now with, again, really one of my heroes, Joan Brugge, and to further look at this. And really when we came across this question, we said, well, this is interesting, this is cool, but it also kind of makes intuitive sense, other antioxidants. Can we take a step back and look at this in a more broader fashion? Is there other stresses that are underappreciated that glutathione could be playing a strong role in? Are there other proteins that could be inhibited? And really, can we look at this on a broader scale? Can we be as comprehensive as possible in understanding the different vulnerabilities the cancer cell is put under when glutathione synthesis is blocked? And so I started doing this. I started looking at this and combining different drugs, blocking glutathione synthesis, and testing a dose response curve against this inhibitor and then this inhibitor. And ultimately I was doing a lot of this, which is a 96-well dose response curve. And this isn't a picture of me, but this very well could have been a picture of me. And it was taking a lot of time. It was, anyone that's done this is quite laborious. And ultimately you hit a wall with saying, you just can't check everything. I was very, very fortunate though that one floor above us at Harvard Medical School was the screening facility, and they did drug discovery. And if you really think of drug discovery and high throughput screening, that is looking at everything. That's looking at as many compounds that you wanted, as there's no upper limit to a large extent. And so then, we started thinking and saying, can we marry these two technologies together? Can we take the high throughput screening, and now instead of using drug discovery, can we more fit it for understanding different drug vulnerabilities? And so this is where we came up with this platform we called MAPS, a multifunctional approach to pharmacologic screening. And so here, unlike drug discovery, we're working with a much smaller number of compounds, but here we're using multiple different doses where we can get a very good understanding of the different vulnerabilities. Furthermore, as compared to large studies from groups like the Broad and the Sanger Institute that do pharmacogenomics, we're really focusing on simply pharmacologic screening, but having a very tight question. Our question was, what are the different drug vulnerabilities if you block glutathione synthesis? But we've gone on to ask many different questions, and trying to see if you perturb one condition, whether it be a metabolic environment, a gene, a drug, what are the different drug sensitivities? So to do this, we had to get a library of compounds. As I mentioned, we were not doing drug discovery. We were actually wanting to work with compounds that we knew had target efficacy. And so we started with a library of cancer compounds. Here is about 400 compounds. This was publicly available from Cell Chemicals, and they really hit a lot of the known pathways. But since we were interested in studying metabolism, we wanted to, make a metabolic inhibitor library. And so here we really started from scratch with the help of a very talented research associate, Jen Endres, who's now with John Blennis at Sloan Kettering. We went through all the literature, gathered these lists and lists of compounds, and their associated targets and their associated pathways, ordered drug compounds, solubilized them, and put them into these high throughput screening plates. And so this we named it our metabolic inhibitor library, and we really felt it, and we have found that it has a lot of coverage of pathways that aren't typically included for cancer-related targets. For our screen, we wanted to actually steer clear of typical cell viability readouts for high throughput screens, namely cell titer glow. Again, cell titer glow works really great in a lot of different situations, but since we were looking at a metabolic pathway and even perturbing metabolic enzymes, we didn't want to have ATP and a metabolite be the readout of cell viability. We could have, in some situations, maybe a few cells making a lot of ATP, or we could have a bunch of cells that are okay, but they're just not making any ATP. And so we relied on an image-based screen where we're simply washing away dead cells, staining the nuclei, and counting cells using a high content scope. And so you can see here, this is just how we count the cells. And if we have a strong treatment, we would lose cell numbers. And what do our drug libraries actually look like? Really just briefly, as I mentioned, there's these 10-point dose curves. It's ranging from 20 micromolar down to either 3 or 1 nanomolar. And again, what we're doing here is we're stamping on this plate, whereas, you know, typical drug discovery efforts, each of these wells would be a different compound at 10 micromolar, a single concentration. Here we're stamping on the dose response curve. And so this really, again, takes any sort of human labor or error out of the equation. And very rapidly, we get this 10-point dose curve. And so, you know, here we can ask our question, again, our question was, what happens when you deplete glutathione synthesis using this chemical compound BSO? And we were looking for, you know, different compounds that could potentially reduce our area under the curve. This is the ultimate metric that we're doing. We're just calculating area under the curve and seeing is there increased sensitivity. And we really think this platform or this approach complements genetic screening approaches and even other screening approaches. And I think there is a space for this to exist. I know a lot of other groups such as Scott Dixon and others have really found great stuff in this space, and because, you know, there's definite advantages that can complement genetic screening. You know, having compounds that hit multiple different isoforms get around target redundancy. Having the dose response allows you to overcome cell essential genes where, you know, with the genetic screen, a full knockout, a typical CRISPR screen, you would have basically a loss of cell viability for these essential genes. Here we can titrate it back. A lot of these compounds are FDA approved, and even if they're not, there's some understanding behind them that they can be translated very quickly to higher order systems. And what I really liked is we get these responses and the data back quickly and at a low cost. And so we've really used this platform a lot going forward. You know, here just measuring the reproducibility, we did this one screen with this breast cancer cell line, SUM149, measured the area under the curve for numerous different compounds, and did the screen also a year before, and it was a very tight R-squared, showing that it's, again, reproducible results. And we've been really fortunate to collaborate with numerous different groups and doing these screens and even sharing these metabolic inhibitor libraries. And so, again, anyone that's interested, please let us know. We're very open to sharing this as a collaboration and providing these libraries. Okay, so back to glutathione, which was our main focus from the beginning. We did the screen and we said, okay, what would happen to drug sensitivities if you inhibited glutathione synthesis? And so we took breast cancer cell line, treated it with or without BSO, and then looked at now the difference in the area under the curve for all these different compounds. So this is the two libraries together, about 600 compounds. And the reduction in AUC, the top hit was auranofin. Now this compound shows efficacy against thyrodoxine reductase. This is a key enzyme in the thyrodoxine pathway. And so this was actually really great to see that this was, pretty much a positive control built into the system. We also got a hit being GLS, a compound Beptes against GLS. Now GLS glutaminase makes glutamate, which is really part of the glutathione pathway. So this to a certain extent made sense, although there's definitely more questions to go along with it. But what really jumped out was a bunch of inhibitors against deubiquinases, or DUBs. And so this was really exciting. It was a different area, and we wanted to pursue it. And all this work actually has been published about a year ago in Cell Metabolism. And so without going into too much detail, we did validation of these compounds. And so now if we pull this out to a growth curve, looking at a fold increase in cell number over time, just trying to see that, we can see really clearly that each of these drugs on their own has very little or no effect. BSO on its own, now in the red curve, solid red curve, has no effect, but combining these together really wipes out the cell. There's a complete loss in cell number, and this is largely due to a massive increase in cell death. And so again, all this work is published. I don't want to get into too much detail behind it, but what we think the potential hypothesis behind this is that glutathione plays a large role in regulating protein homeostasis and buffering both ER and proteotoxic stress. And deubiquinases are really part of this pathway. And so if you block glutathione along with deubiquinases, you get this massive increase in ubiquinate proteins. This is what deubiquinases do. They deubiquinate proteins. And so this massive increase in ubiquinated proteins ultimately leads to proteotoxic stress and ER stress and cell death. And so we think that using this high throughput screening approach has kind of uncovered some really interesting and new biology and stuff that we can potentially take forward into even preclinical trials or different therapies. Now, in the last part of the talk today, I'm going to be focusing on a bunch of new data. And so this is all unpublished data. We're really excited to, with this project. And this is really taking a step back and saying, what are the roles of glutathione in just normal cells? This is a factor that's been known for more than 140 years. But what do we actually know about this in vivo? And if we can know more about this in just normal cells, can this better inform us of how cancer cells are potentially using glutathione? And a lot of this stems from some problems we had with very early studies. And this was basically using this chemical compound BSO. And so for our initial studies and trying to inhibit glutathione synthesis at different time frames, we used BSO, but we really knew that, especially for in vivo delivery, that treatment with this chemical compound wasn't perfect. It was far from that. It had very poor pharmacokinetics. It gets cleared from the plasma quite rapidly with a half-life of about 1 hour. And on top of that, even if we were to continuously treat mice or deliver mice with BSO in vivo, not all tissues were losing glutathione. And so this was a published report from quite some years ago now, where they did this, measured the area under the time concentration curve as a readout of residual glutathione. over time. And what they found was a lot of tissues had a high amount of glutathione left, most notably the liver, where it's almost 50% of the glutathione is left. And so, these sort of issues compounded with, just the general, even overall scientific literature, was that, alterations in glutathione and oxidative stress is found in almost every disease. It's found in aging, it's found in cancer, it's found in inflammation, and the list goes on. But whether these changes are causative is not explicitly clear. And we thought one way we could actually start to approach this is saying, instead of looking at glutathione changes at the end, let's start with the changes in glutathione. Let's make this being the initiating insult, and let's see what would happen. And really, let's look throughout all the tissues, and let's look at this in vivo. For a long time, though, this was not possible because GCLC is actually an essential gene for embryogenesis. It's the straight knockout is very early embryonic lethality at E6.5. And so to circumvent this, we decided to use an inducible Cree system. And so here we skipped embryogenesis and went all the way to adulthood to say, okay, if we have an adult mouse and then induce the deletion, now using this flox GCLC and a ROSA 26 Cree ERT2. And so this is Cree expressed everywhere, but in an active form where it can be induced by treatment with tamoxifen. So now with this inducible model, we said, okay, can we ask this question of what glutathione is doing in adult tissues? And really, again, this was motivated by previous studies, not only by our group, but others. We're seeing that necessities of antioxidants and even other metabolic factors changes over time. That context is extremely important. And so this is why we thought, this is potentially, again, it's very naive approach, it's very open-ended approach, but we thought we could potentially learn some really useful information from this. And so we did it. We collaborated with Vasilis Vasilou, who had the GCLC flox mice. We crossed them to the ROSA 26 Cree ERT2, and we treated the mice with tamoxifen. So our control are the flox with tamoxifen, comparing the flox with the inducible Cree with tamoxifen. And again, the tamoxifen becomes 4-hydroxytamoxifen, and that induces the Cree, where you would get excision of the GCLC. We first honed in on a couple organs, the liver, kidney, lung, and brain. And one tissue we were very much excited to look at was the liver. This is because the liver is the largest producer of glutathione out of all the organs. And so we wanted to make this one of the tissues we were looking at right from the get-go. If we look over time and harvest tissue, what do we actually get a deletion? We do. Now looking at mRNA levels across these organs, we get very good deletion in the liver, very good deletion in the kidney, less so in the lung, and far less in the brain. And this is potentially due to the ability of tamoxifen to reach the brain tissue. But this alone is still very interesting. We haven't followed up on this because it would hypothetically be 50% of these cells isolated from the brain would have a full deletion of GCLC. But we really focused a lot of our work on some of these other tissues, and now just going forth with the liver, looking at the protein. GCLC protein is completely lost within these liver tissues of these knockout mice. And now we collaborate with a very close collaborator, Gina De Nicola, her postdoc Pio Kang at the Moffitt Cancer Center in Florida, where they really have an expertise in doing metabolomics and, even more importantly, measuring these redox cofactors to ensure differences and trapping them within their reduced or oxidized state. And so here, measuring glutathione levels, we get a very robust, more than 90% depletion of glutathione within the liver tissue. So what happens? You know, this is, we had confirmed to a large extent that the tool was, you know, that the model was working, but what actually happens? And so if we do just simple transcriptomics and RNA-seq, we see a very nice induction of oxidative stress gene signatures. So these are a lot of targets of NRF2, and oh, there's chat. Oh, these are targets of NRF2 and other factors, but again, it was very encouraging to see that there was a strong oxidative stress gene response. Oops. We also saw genes related to ubiquination, protein homeostasis, and ER stress. And so this again fell in line with our previous studies that glutathione was playing a very strong role in buffering protein homeostasis. But what actually happened to these mice? And so this was the thing we were most interested in. And what was quite interesting was that these mice started to lose weight fast. And by fast, I mean within 2 weeks, they had lost more than 20% of their body weight. And they were just progressively losing their body weight over time. Now, our first thought was, okay, maybe these mice are just undergoing organ failure, especially from something like the liver, we were really banking on. But it was surprising, though, that even though these mice were losing weight, they were still grooming themselves, they were still running around, and they were still actually eating food. And so here, if we measure food consumption over time, there's actually no change. There's even a little bit higher at some time points of the knockout. And so it wasn't that these mice were not eating food, even though they were losing all this weight. And so we, you know, our next question was, well, where is this weight loss coming from, or how do the organs look? And so we looked at the organs we had seen before, or looked at before, liver, kidney, lung, and brain. We saw a very representative reduction in total mass across these organs. We take this to other organs, we see this as well, intestine, spleen, pancreas, stomach, heart. But what really jumped out was the fat tissue. And so looking here at epididymal gonadal white adipose tissue, these mice had lost pretty much all of it. There was basically no white adipose tissue left in these depots. And if we look under histology, this is a very dramatic effect. And so here, you know, in a wild type mice, we get these very nice adipocytes that are really filled with fat. Whereas in the knockout mice, again, this is the same magnification. These are greatly shrunk, and these fat depots are largely dissipated. And so some of our questions going forward now with this major phenotype is, well, what is behind it? What is behind this fat loss and this overall body loss? And we've kind of taken two different approaches. Is it that the mice are not making enough fat? Is there not enough anabolism or production of the fat? Or is it a catabolism issue where we're just burning fat at a higher rate and potentially a disruption in nutrient absorption? And so I'll just to show a couple hypotheses on this. The first, hypothesis that we had was that this was related to adipose generation, and this was really because when we draw out these antioxidant pathways, they're actually very much connected to other metabolic pathways. And so one factor that plays a role between these two processes is NADPH. And so NADPH, it's synthesized through multiple different metabolic pathways, and it's a key enzyme, or sorry, it's a key cofactor that works through these enzymes, glutathione reductase, thyrodoxine reductase, to regenerate these antioxidants. What's really interesting, though, is NADPH is a cofactor for numerous different processes, including fatty acid synthesis. And so it's required and used by FASN to generate new fatty acids. And so we had, again, a very kind of naive hypothesis was, if we simply stop the production of glutathione here with the deletion of GCLC, would there be an increased reliance on regenerating antioxidant species and using NADPH to do this? And so, especially in the case of thyrodoxine, where its main regeneration comes through NADPH, would we be getting almost a shift in the balance of utilization of NADPH away from fat synthesis towards antioxidant regeneration? You know, this is, again, really preliminary data. Unfortunately, we due to the disruption in research, we haven't had a chance to chase all of this down. And again, I've just started my lab at, I think I mentioned at the beginning, or maybe I didn't, at the University of Rochester. Our lab is quite new. And so we haven't had a chance to chase all this down, but even just looking at our previous data and our high throughput screens, if we actually treat mice with BSO, we get and inhibit glutathione synthesis, we do get increased sensitivity of cancer cells to an inhibitor of fatty acid synthase, cerulenin. And we also conducted a CRISPR screen looking for genetic targets of sensitivity upon glutathione depletion. And so here, looking at relative abundance of sgRNA guides over time, either comparing a non-treated or cells treated with BSO, we see that the cells that contained sgRNA against FASN were much less abundant, suggesting that, again, inhibition of FASN in the presence of glutathione depletion is much more sensitive. And so this was the idea of possibly this weight change and this fat response was due to a lack of production of the fat. But we also wanted to just go kind of digging and see what are happening to other tissues that could potentially explain this, and even potentially touch on other phenotypes. And so when looking at this, what really jumped out was the bone marrow tissue. Here in a representative slide of the knockout, that you get a very large amount of loss of cell numbers within the bone marrow. This actually is in line with numerous other studies of connecting oxidative stress to aberrant differentiation and within the hemopoietic system. So this was really exciting. We're excited to pursue this more. But with regards to the whole body weight loss phenotype, one organ that really jumped out was the intestinal tissue. And so we were looking at this and we found that these mice were undergoing enteritis and colitis and severely disrupted villi and colonic and crypts. And if we go down to the colon, what we were seeing was that these large abscess crypts were being formed shortly after or within this time frame of the weight loss and the deletion of GCLC. And so one hypothesis that we have is that glutathione is very integral to intestinal stem cell homeostasis. Loss of glutathione could potentially be disrupting these tissues, leading to malabsorption of nutrients and therefore catabolism and breakdown of different tissues to try to maintain energy supply. And so this again really branches into different physiology, but even bringing it back to cancer, you know, is this a tissue that might be much more sensitive to perturbations in glutathione, and really why is it the intestinal tissue? And we really have a lot of questions about this, and we're really excited to pursue this further. And now finally, one tissue that really we were banking on to have an effect that didn't was the liver, and this was really interesting. You know, the liver was the largest producer of glutathione out of all our tissues. It definitely had been shown to play a very strong role in detoxification of toxins, but in the absence of toxins, under normal just homeostasis and just resting conditions, we were finding that the liver could lose all this glutathione and be completely okay. There was no histological and no dramatic histological changes. There was no biochemical changes in terms of ALT, AST. There's no liver steatosis. And so this has really started to make us think, well, where is this glutathione going? It's making all this glutathione. Under most conditions, it might not need it. Where could it be going? And I'll touch on that just in a couple slides, but we're really excited to now look at how the liver and its production of glutathione could be contributing to other systems. And now just the last piece of data, again, not to show, have too much in this presentation, but we wanted to look at this, try to use this genetic model and to take it one step further to look at tumor cell intrinsic deletion of GCLC and glutathione synthesis. If you remember my very early studies, the very early studies that we did with GCLM, this was a systemic reduction in glutathione synthesis. When we treated with BSO, this was, again, a systemic reduction in glutathione synthesis. What I wanted to really look at was, especially and fully confirm the lack of sensitivity to glutathione depletion in these tumors. What we did was we crossed these ROSA 26 Cree ERT2 mice to now a PYMT GCLC flox mouse. And so here these mice get tumors, they get breast cancers, but we can actually transplant these tumors to a host recipient. These are all under the Black 6 background, so there's no problem with host having to use an immunocompromised mice. These tumors grew great. And now if we actually treat with tamoxifen, we're getting a tumor cell intrinsic deletion. And so we wanted to look at this and really nail this down to see what happens if you tumor cell intrinsically lose glutathione synthesis. And so we set up this model. It took quite some time, but we got it going. We treated with tamoxifen. We get a very nice reduction in GCLC transcripts, very robust reduction in glutathione levels. Again, this work was done by Gina De Nicola's group and looking at these metabolites. And what was quite, again, dramatic was that these tumors were largely unperturbed. And so again, this is very preliminary data. We obviously want to follow up on this and repeat this and look possibly in different tumors and different scenarios. But with these breast tumors, they were largely unperturbed. And so there was no difference in tumor volume, no difference in tumor mass. And so this really, along with even the liver tissue, started to make us think more and more about, okay, what is glutathione doing outside the cell? And maybe its extrinsic synthesis or its synthesis by other tissues is very important for normal homeostasis and even cancer cell survival. And so one hypothesis that we're trying to chase down now is that really trying to hone in on extracellular glutathione levels produced by tissues such as the liver to supply it into the serum and really the catabolism of extracellular glutathione. And so it's really driven by this one extracellular enzyme GGT1, this transferase that breaks down glutathione. And now in mammalian cells, it has not been reported to a large extent that you can actually get uptake of glutathione. It actually has to be broken down into its amino acids, glutamate, cysteine, glycine, and then these can be taken up. And so we really think that circulating glutathione is this large pool of metabolites that potentially have not been fully explored in terms of a metabolic vulnerability for cancer cells, and even to better understand normal physiology with metabolism. And so we're really excited to further investigate this and really chase this down. And so with this new project, just to wrap up, we really have a lot of different paths to take this. Is there a connection with weight loss and glutathione, specifically with regards to fasting, cachexia, because these mice lose weight so rapidly? What exactly is going on? We're excited to chase this down. Is there a problem with nutrient absorption in the intestinal tissues? Where is this necessity of having glutathione in the intestinal tissues coming from? We have some ideas with regards to this, particularly, is it the microbiota and really putting this oxidative stress onto the cells and placing them in this requirement for glutathione synthesis. We'd obviously like to follow up on the bone marrow phenotype with regards to loss of glutathione. Again, these phenotypes have been explored with regards to adding reactive oxygen species, in the forms of radiation or other insults, but we're really excited because here we're getting the endogenous buildup of oxidative stress and the endogenous buildup of ROS. And so, even to just ask old questions with new technology to fully go into what are the impacts of antioxidants and oxidative stress on hematopoietic system and differentiation. And finally, following up on the liver, in the absence of toxins, what is the body doing with all this glutathione? And is it really going into the serum? Is it supplying a metabolic resource to numerous different tissues, including tumor tissue? And really, with a lot of this work, we'd like to bring it back to cancers and better understand how we can potentially perturb these pathways to have a therapeutic benefit for diseases such as cancer. And so this is really the focus of my lab moving forward. Again, we're a very new lab, but we love collaborations and new insights, and we're looking to grow. And so again, we're focusing on using two aspects of these two technologies, being mouse modeling and screening approaches, high throughput screening approaches, to better understand these roles of antioxidants in cancer. There's a lot of people to thank. My lab is, again, quite tiny, but we have really great people. Lab manager, Haley Chang, undergrad student, Catherine Rodriguez, and our rotation student, Emily Witt. We've had great support from our screening facility at URMC, University of Rochester Medical Center, John Ashton, Marlene Bayless. We have great collaborations with investigators there, Josh Munger and Stefano Mello. And really, I've been extremely fortunate to have numerous different collaborations over the years. My previous supervisor, Tak Mack at Princess Margaret, Joan Brugge at Harvard Medical School, Gina, Alessandro, Dirk, and really numerous others have made it made it possible to actually ask these questions. And so I'm really excited to continue to ask these questions moving forward and really collaborate with others to try to get new insights into this. And so hit us up on Twitter or check out our lab webpage. Obviously, thanks again to Philip for providing this opportunity to present our work and present our new projects. And yeah, I'm excited to answer any sort of questions via Twitter or email or anything going forward. So that's about it. I think I'm going to stop there, and I don't know if we'll be turning it over to Philip, but again, thanks a lot, everyone, and I will talk to you soon.